Okay. How you doing, folks? Today I'm going to draw for you a bacterial cell. And this cell I'm going to draw for you is going to be a bacillus, also known as a rod. So it's not going to be a coccus. It's not going to be a spirillus. It's not going to be a sphere or a disc shape. It's going to be a hot dog shape or a rod. Okay, so when we're drawing that, this is going to be a generalized bacterial cell. So you're probably never going to see a cell exactly like this one, but it's going to have a lot of the parts that you might expect to find on your generalized cell. So things you might encounter. All right, so it's going to be hot dog shaped. So let's go ahead and draw kind of a hot dog shape. All right. So already we've got two things that are found in all cells. So this is going to be our plasma membrane here. And the plasma membrane, that's something that surrounds every single kind of cell, whether it's bacterial, archaeal, eukaryotic. All cells have a plasma membrane. All cellular life has plasma membranes made up of phospholipids and proteins. And that plasma membrane forms a boundary around the cytosol or cytoplasm. What's the difference between cytosol and cytoplasm? Cytosol is more generally applied to prokaryotic structures because it's a cellular solution, whereas the cytoplasm suggests that it has more structure to it, like, for example, having things like a cytoskeleton, which is something we don't see in bacterial cells. We only see cytoskeletons in eukaryotes. Although there's some evidence that there's some oddball archaeans that may have a rudimentary type of cytoskeleton. Okay, but that's not our story to tell right now. We're just talking about the cytosol or the cytoplasm. We'll kind of use those terms interchangeably. Okay, so every cell's got a plasma membrane. Every cell's got some type of cell sauce or cellular solution, cell stuff inside. Okay, so next I'm going to draw another thing that's kind of somewhat sort of universal, and if you're drawing along with me, it doesn't really matter what shape you're going to draw. Essentially you just want it to look like a big twisted up loop of stuff, and that's what it is. This is the chromosome or the DNA of the cell. And typically we find it in a bacterial cell scrunched up in one part of it, and we call that one part of it the nucleoid. We call it the nucleoid because we're eukaryotes, and when we see something that looks like a collection of DNA, we think it looks like a nucleus, even if it doesn't have another membrane surrounding it. So even though this type of arrangement of having DNA has been around for a lot longer than the nucleus, when you're a hammer, everything looks like a nail. And when you're a eukaryote, every mass of DNA looks kind of sort of like a nucleus, even when it is not bounded by a nuclear envelope. Okay, so DNA is present in all cells, but a nucleoid, that's a prokaryotic type of thing. Okay, what else are we going to find inside? I'm just going to draw these things as little circles. If we were to zoom out on one, we'd find that we've got ribosomes, which are made up of a large subunit and a small subunit. Now, I'll go ahead and label those little dots there, point specifically at one, and call it a ribosome. And ribosomes are something that we see very similar structure, be it in a prokaryotic type of cell or a eukaryotic cell, in terms of having the large subunit and the small subunit. But the sizes of the two subunits overall is different between the different domains of life. So bacterial ribosomes, you can think of them as looking more like a sesame seed bun, and a eukaryotic ribosome looks more like a pretzel bun. It's a little bit bigger, but still works pretty much the same way in that it's got the two sections to it. So it's got a large subunit and a small subunit. This is important because many of our antibiotics function by targeting being competitive inhibitors of the AP and E sites within the large subunit of the ribosome. So if you eat a bunch of clindamycin, clarithromycin, zithromycin, that family of antibiotics, the way it functions is by blocking these the active sites in the large subunit. So you can eat that, and it's not going to affect your personal ribosomes in your cells because they're a different size than the ones in your bacteria. But you can also damage your beneficial bacteria as well as any potentially pathogenic ones, which is one of many reasons why you shouldn't just take antibiotics constitutively all the time. Okie dokie. All right, let's see. What else do we have on the inside? Oh, I'll draw one more thing on the inside, and I'll draw it down here for reasons that we'll get into in a minute. This is a plasmid. And plasmids are loops of DNA that may be found inside the cytoplasm of a bacterial cell. So in addition to the main chromosome, which is in the nucleoid, we might have other little loops of DNA. And I think of this, the DNA of the nucleoid being kind of like the operating system that has all of the stuff that is absolutely necessary to make the cell run, all of the housekeeping genes and things like that. Plasmids are kind of like add-ons. They're kind of like applications that may give some type of ability to the cell that not all cells are going to have. So plasmid, call it a loop of DNA. And an example of something a plasmid might have encoded on that loop of DNA is something like antibiotic resistance. So maybe this particular cell with its plasmid can grow in the presence of antibiotics like penicillin. Penicillin resistance is a problem, and one of the reasons why penicillin isn't as widely prescribed as it was when it was first introduced as a modern miracle drug. Okay, so that's most of the inside stuff. Fairly simple. No membrane-bound organelles, no mitochondria, no chloroplasts. I'm going to go ahead and draw for you now one single flagellum. One single flagellum. If we had more than one flagellum, we'd have the plural of one flagellum, our flagella. Because it does come to us from a Latin word. So one flagellum, two flagella. Both prokaryotic cells and eukaryotic cells have flagella. However, this bacterial flagellum is very different from a eukaryotic flagellum. And what I mean by that is a eukaryotic flagellum is powered by microtubules, which is something that prokaryotic cells don't have as part of the cytoskeleton. So a eukaryotic flagellum I think of as kind of being like the tail of a cat or a dog or a mouse or any vertebrate with a tail, where the microtubules are kind of like the bones and the muscles that are inside the tail, and then it's got a sheath of integument, which is the plasma membrane in the case of our flagella, or skin and hair like you would see in a cat or a dog's tail. Whereas the flagellum of a bacterium is more like a weed whacker, in that you've got a filament or a thread, which in the case of the bacterial flagellum is made out of protein. It is not sheathed in any way, not covered like its skin and hair or any its analogs in at the cellular level. But there's a little motor at the base, a little motor protein that is spinning around and flipping that flagellum around to basically do the same thing that our flagella do, which is provide some type of locomotion. Okay, so you've got a motor at the base that's spinning a filament, so it's much more like a weed whacker than it is like, you know, they can't really wag their tails in the same way that like a cat or dog can, you know, especially like a cat tail, you know, which can have a, you know, they can wave it in different directions and things like that. All right, so that's most of the inside stuff. Now we're going to draw some of the outside stuff. So one of the things I need to do here is I'm going to draw another bacterial cell, another hot dog over here. And we'll just say that it's kind of a little bit behind the plane of the page. It's further away. So we're going to call this another cell. And this structure that's connecting them together is called, all right, so I always call it a pilus, but if there's more than one, I call them pili. I don't call them pili. I don't call one a pilus, a pilus. I don't know. Anyway, so what a pilus does is it acts kind of like a straw. And actually the name Pilus means like a straw. Like a drinking straw, because it's hollow. And one thing that can be done with a pilus, which is the normal thing for it to do, is it can take copies of plasmids and transfer them from one cell to another. So it's almost like they're sharing recipes here. It's like other cells like, ooh, I've got a recipe for penicillin resistance. Would you like this other cell? And the other cell's like, well, we're growing in a penicillin-rich medium. Yes, please. I would like that so I can survive. So this process is called conjugation. Once upon a time, I've heard pili referred to as a sex pilus because it is kind of exchanging genetic information, but prokaryotic cells don't have sex. It's not sex. That's something that's very different. All right. So that's one of our structures that we're going to see that can be produced on the outside. Another thing we're going to see outside of the plasma membrane, so we're not really in the cell proper anymore, but I'm just going to try and as carefully as I can just draw another hot dog around my first hot dog. So now I've got a second layer. So external to the plasma membrane, we've got a cell wall. Okay, and the cell wall in our bacterium is going to be made out of a material called peptidoglycan, which you might guess from the name is made up of protein, peptido, and carbohydrate, glycan. So it's a polymer of proteins and carbohydrates that make up this kind of rigid wall. Now, the plasma membrane is kind of soft and malleable and fluid. The cell wall is rigid and hard and has more of a protective function and gives kind of like a rigidity to the cell. Now, you might not see this exact arrangement of having the cell wall like this. Your results may vary depending on whether you have gram-positive or gram-negative bacteria. It's not something I'm going to get into in this particular little chat, because I'm kind of wearing on here. But it's important to know that the majority of bacterial cells have a cell wall that's made out of peptidoglycan. In archaeal cells, because they were discovered later, it was found that their cell walls are made out of a different kind of stuff, which has the rather uncreative name of pseudopeptidoglycan. Again, since we knew about peptidoglycan in bacteria first, archaea kind of got the shaft, and it's like, well, you're peptidoglycan, your outer wall of protein and carbohydrate is the fake one. This is the real stuff. It's chemically different. I can't exactly tell you how, but it is. And that's, for our purposes, that's all we need to know. Okay. Let's go ahead and we're going to draw one more layer of hot dog outside of our plasma membrane and outside of our cell wall. Being very, very careful. I'm doing a pretty good job, if I do say so myself. And this third layer of hot dog is another layer of stuff. This stuff is called a capsule. And unlike the cell wall and the plasma membrane, we don't always see a capsule in bacterial cells. One place where we particularly see them is in symbiotic cells, pathogenic cells, cells of organisms that cause disease. Because what the capsule does is it's made out of a polymer of polysaccharide. Which helps the bacterium to stick to its host. Now, bacteria, being as small as they are, they don't experience gravity in the same way that you and I do. So if bacteria are inside your body, they don't just all kind of drift down to your feet because that's the closest part of you to the ground. They're going to stick to whatever part of your body they can. So, for example, Streptococcus, one of the causal agents of a strep throat, wants to stick to the back of your throat. And it can do that using capsule. And the capsule has a consistency that's kind of like, it's sticky and gooey, kind of like a particular sticky, gooey polysaccharide that we, as higher vertebrates, produce, which is mucus. But we actually use the mucus to kind of move the bacteria and other nasty stuff away, and they use their capsule to try and stick to whatever their host organism is. Okay, let me see. I think I've got at least one more thing, maybe exactly equal to one more thing that I'm going to draw on here. And I'm going to draw it kind of look like it's kind of got a crazy... Like it's just received some shocking news, this bacterium, and it's been frightened. So what are these little hair-like things that I'm drawing, studding the outside of this bacterial cell? Well, I'll tell you what they're not. They're not cilia. Cilia, as you may or may not recall, but you should know now that I'm telling you, cilia are also powered by microtubules, like a eukaryotic flagellum. And as I said, bacteria don't have a complex cytoskeleton. So they do not have cilia because they don't have the microtubules to power them. They don't have hair either. Hair is something that we only find in mammals, in vertebrates. Hair in the sense of the same chemical composition and all that stuff. So what are these things if they're not hair and they're not cilia? They are a thing that, if we have just one, it's called a fimbria. If we have many of them, plural of fimbria is Fimbri, not Fibri. Come on, squeeze all in there, all you letters. So we added just E to the end to make several Fimbri. And what Fimbri do is they have a similar job, though not a similar structure, to the capsule. Fimbri can kind of help to anchor bacterial cells in place so that they can do what they need to survive and reproduce. They can adhere to host tissue, things like that. Okay, so I'm looking over my picture that I'm actually copying to make this one. And I think I've got all of the pieces in there that I have in my first picture. So there you've got your generalized bacterial cell, your bacillus, with all of these special parts that, again, you're never going to see a cell exactly like this under a very powerful microscope, because these are teeny little cells. But you might see some complement of all of these parts when you're looking at bacterial anatomy. So metabolically, bacteria way more sophisticated than eukaryotic cells, but morphologically, just from looking at the structures that are present, pretty simple. All right. Thank you for watching and hopefully drawing along with me.